Yeah, okay. My lungs are not enough. Get a few inhalers. Maybe steroids. Yeah.
All right, so let's get started. A couple announcements before we go here. Enough of you have said something to me that makes me think that I must have made a mistake somewhere in lecture. That I must have said that prokaryotes don't have a cell wall. Mostly they do. They don't have a membrane-bound nucleus. That's what makes them prokaryotic. I went back to lectures and I couldn't find where I said that wrong anywhere, but there are a few prokaryotes that don't have a cell wall. That is the rare exception. That's the mycoplasma. But you know, we went through the gram-positive cell wall, the gram-negative cell wall, the acid-fast cell wall. So the overwhelming majority of the time they do. Okay, they just don't have a membrane-bound nucleus. So just clarify that. Also, I'm still getting a few of you asking about homework questions and say, I got it right, the computer marked it wrong. Yes, I have to hand grade all those fill in the blank. And there's also some kind of short answer type questions as well that I look at very briefly. So that is easier for me to do after the due date. So I'll start working on that on Wednesday for that first round of homework. And remember, our first exam is Wednesday. So if you have any questions, I will make known today, you know, the exam covers until right now. So I'm hoping that we can get through that pretty easily, but we'll have to keep moving here. So, all right. So yeah, plan on being here Wednesday. I think you'll have plenty of time to do it. It's a shortened exam. I'm going to give you 50 minutes, and then we're going to do about 15 minutes of lecture, 15, 20 minutes after that. So. Okay, so we were going through prokaryotic cell structure. So this is in chapter 3. Hopefully I know some of you haven't started your homework yet. So if you haven't, it's time to start working on that. So it's not as painful as what it needs to be. We've been working through the prokaryotic cells, mostly thinking about bacteria. We're going to talk briefly about some differences with the Archaea, but we're mostly thinking about bacteria. And we've kind of working from the outside in. We started with the Glycocalyx, which is really a general term for either a capsule or a slime layer. A lot of times we just call it a capsule, but there's kind of a general term for that. Working in from different like appendages like Flagella or Fimbriae. We spent a good bit of time on cell wall structure. And then we were talking a little bit about the cell membrane and finished up kind of thinking about osmosis. If that, I kind of stumbled through that a little bit. I apologize if that is something that's confusing to you. Make sure you get straight on that. Come talk to me about that. That's one of those big concepts in biology that you as a science college graduate should master. So those terms, remember, talk about the solution that the cell is in, hypertonic, hypotonic. Okay, and then water is always going to move from a high concentration of water to a low concentration of water. Okay, so thinking a little bit more about transport through a membrane. Remember, if it's some kind of passive process, that means that no ATP is required. If it's an active process of transport, then ATP is required. It has to spend some energy in order to do that. And I don't really need to get too caught up into these different names like uniport and antiport, whatever. The main idea here is that there's some kind of carrier protein that's necessary in that plasma membrane in order, and again, ATP has to be spent to bring that, whatever, if it's a nutrient or whatever, into the cell. So again, a uniport, just kind of that protein channel is one direction. With the antiport or the coupled transport, basically it's just a little bit more energetically favorable, maybe for something to maybe diffuse out, like an ion or something like that, so that, you know, whatever that thing is that the cell really needs can be transported in. There is one additional kind of transport that only prokaryotes can do. Okay, so this is unique to prokaryotes, and that's called group translocation. So with group translocation, whatever the cell is trying to bring in, that's going to be modified at the same time that whatever nutrient or whatever is brought in. And the best example that I have of that is something that happens, it's the first step of glycolysis. Okay, so the cell changes glucose to glucose 6-phosphate. Okay, that actually happens when the glucose is transported in the cell, that phosphate group is added. Why would the cell want to do that? It's really an ingenious way. If the cell is trying to get, lots of glucose kind of concentrated inside of the cell, okay, it doesn't want to lose that glucose. It doesn't want to lose it by diffusion, right, out again through the membrane. So if it modifies it a little bit and changes it to glucose 6-phosphate, then it won't diffuse right back out again. Okay, so it's a way to concentrate the glucose inside of the cell. Okay. And again, this is just something that bacteria do and not our cells, apparently. Okay, so that's pretty much kind of the outside of the cell. We're ready to move into the prokaryotic cell. And it seems like we spend a lot of time on the outside. There's kind of much less going on inside. So not to worry too much. Okay, so internal structures. The cytoplasm and the cytosol are two terms that probably you would think about being fairly interchangeable. And a lot of times they are. If you use those terms absolutely correctly, when we refer to the cytoplasm, that refers to anything basically enclosed by the cell membrane. So that would include, if we were talking about a eukaryotic cell, that would include any organelle. Like technically, the lysosomes are part of the cytoplasm. And then the cytosol is the fluid present inside of the cell. So that's the fluid cytoplasm that kind of fills in all around the internal structures. Okay. For the prokaryotic cells, they have a nucleoid region, and that's where the DNA is located. Okay, so there's no membrane around there. It's kind of just a region of the cytoplasm, basically. And we'll talk more about how that DNA is stored for bacteria later on, but typically that involves a chromosome. Okay, that doesn't look right. And bacteria usually have one or two, most often one, sometimes two, and that's in a circular form. And it's haploid. Okay, so kind of a big long, circle like this, maybe. And then most bacteria are going to have lots of different plasmids as well in that nucleoid region, probably lots of them. These are also circular, but kind of small circles of DNA. And plasmids usually encode for information that's not absolutely necessary for the cell to survive. Okay, so all of your cell structures, all of your maybe important enzymes for metabolism are going to be encoded on the chromosome, and then maybe something that would help the cell survive in a particular situation, like resistance to an antibiotic, is probably, that information will probably be on a plasmid. But I'll give you more examples of that later when we get into our section on genetics. All prokaryotes as well as eukaryotes are going to have ribosomes. So I thought, well, let's just talk about both the prokaryotic form and the eukaryotic form together here. Remember, I think I mentioned this before. I know I mentioned that this is a site of protein synthesis. We'll talk more about that later. Also, I think I mentioned that these are not membrane bound. Okay, so here I was researching this because it wasn't really making sense to me. Technically, ribosomes are called organelles, and organelles are defined as membrane-bound structures, but the ribosomes don't have a membrane. So sometimes definitions don't totally make sense. You can call them an organelle if you want, but they don't have a membrane around them. They are going to be composed of ribosomal RNA and proteins. And there's actually two different related but different structures. Here's the prokaryotic ribosomes on the left side and the eukaryotic structure on the right. So you can see both the prokaryotic and eukaryotic structures have a small subunit and a large subunit. They will come together during protein synthesis around the messenger RNA and make one complex. Okay, so that's why it doesn't really make sense that they're membrane bound, that they would be membrane bound. Okay, the size is actually measured in a unit S, which means Svedbergs. That's what this S stands for. So the small subunit here is size 30S, the large subunit 50S, and those come together to form a 70S ribosome. Now, if your math's good, you'll notice that that doesn't really make a lot of sense. Okay, this unit, Svedbergs, is actually a measure of sedimentation rate. So basically, when you would spin ribosomes down in a centrifuge, it's how quickly they're going to descend to the bottom. So it's the sedimentation rate is kind of a measure of weight and kind of overall shape, kind of the size. Okay, so it's not a linear measurement. So that's why when we have the 50S and the 30S, it doesn't necessarily, it equals 70 instead of 80. Okay, the eukaryotic ribosome, large subunit 60S, small is 40S, and that adds up to 80. Okay, so Svedbergs sedimentation rate. Okay, I said that it was made of RNA and proteins. So the little bits of RNA are the little red lines there. This 16S ribosomal RNA, that's the segment that Wose and Fox sequenced in order to come up with their taxonomic scheme of domains. Okay, so again, just those ribosomal RNA and then lots of proteins in there. Okay. And because the prokaryotes and the eukaryotes have slightly different size, we actually have antibiotics that will capitalize that. We have antibiotics that will target prokaryotic ribosomes and don't have an impact on our eukaryotic ones. So we get some selective toxicity there. And incidentally, the archaeal ribosomes are considered 70S, but they have some different kinds of proteins that make them a little bit unique. But we consider them most like like the bacteria ones. So that's why we can call it prokaryotic. Okay. And then some structures that some bacteria have and some don't. Some can have inclusions. So inclusions are just storage. They function in storage. So in times of plenty, when a bacteria can kind of store a bunch of, some kind of, solute or some kind of nutrient that may not always be available. Okay, so that could be some kind of lipid, starch, phosphates. There's lots of different kinds of inclusion molecules. My favorite one is called polyhydroxybutyrate, or PHB. This is we'll call it a starchy polymer that some bacteria, they can be manipulated to store a whole bunch of polyhydroxybutyrate. And that's what this picture is right here, especially if we grow them in certain kinds of media. I think medium that's low in nitrogen, maybe. And we can get them to fill up like 80% of their cell with this PHB. And we can actually use that to make biodegradable plastics. Okay, I'll come back to this slide, but here's an example of some plastics made with PHB, this bacterial product that we manipulated them to make. And a normal plastic bottle would degrade in the landfill in like 1,000 years, right? But the PHB ones will degrade over a matter of weeks. So that's kind of exciting for some applications. Maybe a water bottle, we don't need it to persist forever. Some things you don't want them to degrade that quickly. But it's kind of exciting. It's more expensive to make plastics that way, but eventually we may be able to get the cost down. So that's kind of exciting. Another example of an inclusion is called volutin. Your book says that has another name, metachromatic granules. But these are made of inorganic phosphate. Cells need lots of phosphate for their metabolism. Magnetosomes, these are inclusions with iron oxide. And some bacteria can use that to kind of help it navigate, kind of a magnetic way to navigate where they may need to go, aiding in their movement. Gas vesicles, maybe you have a bacteria that's photosynthetic and it needs to orient in the water column in just the right place so it gets the adequate amount of sunlight. Okay, so that's an example of a gas vesicle. Okay, so there's other examples. The figure in your book here, that first picture A, that's a picture of the PHB. B there, those are the volutin granules, and C is some sulfur inclusions. We didn't talk about that one. E is the magnetosome, so there's lots of different ones out there. Another structure, kind of wrapping things up here, I think this is the last one I'm going to talk about for prokaryotes that may be present, are endospores. And this is not common in the bacterial world. There's really only a couple of genera. There's two that are really famous for being able to make endospores. That's the Clostridium and the Bacillus. We'll talk more about both those organisms later on in the semester. Some really cool infections that they can cause, like anthrax or C. diff, for instance. And you'll talk more about this in lab. Next week, we're going to do a special stain to be able to see endospores. But this is a structure that we'll call it a survival mechanism. We don't want to mix endospores and bacteria up with sporulation and fungi. In fungi sporulation, you get hundreds of fungi from one cell or one organism. And so that's reproductive. But in bacteria, we end up with one spore from one cell. So this is not reproductive. It's just a way for it to survive in case, the conditions get really bad. Okay, so organisms that are able to sporulate, they're usually present as vegetative cells. So that means metabolically active cells. Somebody said to me once, yeah, but when people are vegetables, they're not moving. Like, yeah, I know, but that's just what they call it. Vegetative cells are the metabolically active ones. And if you look at kind of the life cycle of an organism that's able to form spores, usually they're going to do this when it encounters some kind of harsh environment. Okay, so ideal environments for bacteria are usually warm, moist places. There's a lot of variability there, bacteria that can live in like any environment, but warm, moist places, like, you know, your feet and your armpits. Okay, there's lots of bacteria that live there. That's why those places in our body don't smell very good. Anytime bacteria are not in a warm, moist place, then that could be considered a harsh environment. So some kind of harsh environment that might be a dry environment. It might be where the pH is high or maybe too low. It might be some exposure to radiation like ultraviolet light. Here's a big one, low nutrient availability. Okay, those are all just some examples. There's many more of harsh environments. So when some kind of harsh environment, temperature swings, especially too hot temperature. But when that environment is encountered, that can cause the vegetative cell to start to sporulate. A spore coat develops around the DNA. The spore coat is many, many layers of tough like proteins like keratin, for example. And eventually the vegetative cell drops off and you get this free endospore that can persist for thousands of years. We don't even know the up end of that. And when the conditions get better again, then it can germinate back into a vegetative cell where it can grow and divide like it normally would. Okay, so again, this is not common, but this is a structure that you could find in some bacteria. Okay, so if you compare the metabolically active vegetative cells to the endospores, that's just a little table that compares them. Both of those genera that are able to sporulate happen to be gram positive. Okay, the vegetative cells can easily be killed, but if they just happen to be in the spore state, sometimes they're going to just kind of go through that sporulation cycle as part of their normal metabolism. Then some cells could eventually survive. Okay, and if we stain these, this is a really cool stain to do. Like I said, this is a staining procedure that if you're in lab, you'll get to do this next week. The spores will look green after you finish the staining procedure, and the vegetative cells will be red. And this is the organism that causes anthrax, has very characteristic kind of boxy shape bacilli cells. It looks very, very unique when you, that looks like a gram stain actually. When you gram stain these kind of cells, the spores don't take up the gram stain very well. So you see this, you know, empty white circle in the middle of the cell. That's the spore. Okay. All right, so that's pretty much structures of bacterial cells. Just to point out a few differences between the Archaea and the bacteria domains. This is just a few. There's some other structures and things that the Archaea can make that we're not going to bother to get into here. One big difference in the cell wall. Remember, we said it's only the bacteria domain that has peptidoglycan in the cell wall. Archaea usually have a cell wall, but they don't have peptidoglycan. Any of the eukaryotes that have a cell wall, they do not have peptidoglycan as well. The Archaea can have something very similar, and it's so similar that they call it pseudo-peptidoglycan, but it's not quite the same structure. And there's a lot of variability there as well. The plasma membrane, we didn't get into the nitty-gritty biochemistry of the plasma membrane, but there's some different kinds of linkages with the phospholipids, different kinds of branching. Sometimes they even have a lipid monolayer instead of a bilayer for the Archaea, so some kind of strange things there. And the ribosomes, like I mentioned before, they have the same size as bacteria. And remember, that's 70S, but there are some differences in the proteins that make up the ribosomes that we won't get into too much there. Okay. All right, so let's go on to eukaryotic structures. I don't spend as much time here just because hopefully you've had this numerous times, maybe starting with in elementary school or 7th grade biology. You talked about these different organelles, and we're just gonna kind of hit those really kind of briefly and kind of compare some of the structures that are the same between prokaryotes and eukaryotes. Okay. In terms of cell shape, there's anything goes. There's all sorts of different things. So we're not gonna kind of dwell on that like we did. We had some terminology for the prokaryotes there. So we'll kind of skip over that. The first thing that we talked about for the prokaryotes was that glycocalyx. And that's usually much more pronounced for prokaryotes, but there are some eukaryotic cells that have some kind of a glycocalyx as well. If eukaryotic cells do have a glycocalyx, then it's going to be anchored to the cell membrane. Okay, in other words, any eukaryotic cell with a cell wall typically will not have a glycocalyx. Okay, so cells with cell wall, then there's no glycocalyx. Okay, so it has some of the same kind of functions as a bacterial cell. This first one, attachment, I could not think of an example of a eukaryotic cell that would need to stick to another eukaryotic cell. So I must have read that somewhere, but I'm going to kind of put that in parentheses. Maybe that was one of the main functions of bacterial glycocalyx or capsule, sticking to some structure or other cell. Yes, some protection against drying out for sure. I think this is more of a big function for the eukaryotes, is just strengthening that cell surface. Okay, especially for cells that don't have a cell wall, that's just kind of strengthen up the cell membrane a little bit. Some of our cells actually have a glycocalyx, ones that line our intestinal tract. So it's just a little bit more of a protective layer. Again, maybe, you know, keeps it from drying out a little bit. Okay. All right, cell wall. We're not going to spend a lot of time on this. Remember, certain eukaryotes have a cell wall, certain eukaryotes don't. The ones that do, fungi, plants, algae, okay, have a cell wall. Not animals, right? We don't have cell walls. Not protozoa either, okay? So we'll get more to that, probably more next time. The composition is so varied that we're not going to spend a lot of time on that. Of course, plants have the polysaccharide cellulose. Fungi very often have something called chitin, although there's some variability there. Usually some kind of a polysaccharide. Protozoa don't have cell wall, like I mentioned, but they do have kind of a proteinaceous covering that we'll get more to later. So kind of same functions, strength, support, shape, protection from differences in osmolarity, osmotic pressure. Okay. Cell membrane, a lot of similarities with the cell membrane, phospholipid bilayer. One big difference is that eukaryotic cells, and of course they all have a cell membrane, that's something that every cell needs, will have steroid lipids or sterols like cholesterol for added support. Cholesterol also has many functions in our cell membranes. but helps keep that membrane fluid. Okay, we think about cholesterol as being a bad thing, but it actually is absolutely necessary. Of course, the cell membrane is a semi-permeable barrier. It lets some things in, some things not. With the eukaryotes that don't have a cell wall, it could also function in locomotion, in movement. Okay, so if there's no cell wall to keep it in that certain shape, a single-celled organism could extend pseudopods and then move by amoeboid motion. Pseudopods extension can also give the cell a special kind of transport called endocytosis. Okay, so in this amoeba here, it's extending pseudopods around a paramecium, it looks like, and it's going to digest that paramecium for some food. Okay, so when those pseudopods are extended around something solid, we call that phagocytosis. We're going to walk through that much more detail later on in the semester when we talk about how our white blood cells do that. Pinocytosis is when liquids are phagocytosed. Of course, when those solids or liquids are chewed up and then they're kind of escorted out of the cell, we call that exocytosis. This other kind of endocytosis, receptor-mediated endocytosis, this is how some viruses gain entrance to animal cells. It's kind of a way that they trick our cells into allowing them entrance. Okay, so here's, I kind of like your book's figure that talks about, again, the difference: phagocytosis, some large solid particle, maybe a bacterium or whatever, is ingested. Pinocytosis, you have, it could be, sugars or things that are dissolved in some kind of liquid that get ingested. And then with this receptor mediated endocytosis, there's going to be some kind of receptors that recognize whatever particle. Again, this could be a virus, and that triggers the endocytosis. So that's how COVID gains entrance to our cells. It binds to the ACE2 inhibitor or ACE2 receptors in our lungs. Right? Again, kind of a dirty trick. Okay, so again, bacteria can't do that phagocytosis. They've got a cell wall that prevents that. Another structure that we spent some time on for the bacteria was the flagella. Right, so the only similarity between bacterial flagella and eukaryotic flagella is that both of those structures are used for locomotion. That's really where the similarities end, because the structure is totally different for any eukaryote that has a flagella. For one thing, they are membrane bound for eukaryotes. There is the one exception for the bacteria. Do you remember the bacteria that have the membrane bound? What did we call that? A membrane bound? Axial filaments. Yeah, it's the spirochetes that have the axial filament. Good. Okay, so that's one exception, but for the eukaryotes, they're enclosed in a membrane and they're composed of microtubules. So the bacteria they were made of the protein flagellin. Microtubules are composed of tubulin. And that tubulin is actually in what's called a 9+2 arrangement. Okay, that's why seems like to me a funny way to write this. But basically those microtubules that run through the flagella, there's 9 pairs that go around like this, that shaft, and then there's 2 in the center. So 9 pairs, 2 in the center, 9 plus 2 arrangement. And remember with the prokaryotes, there was a filament and a hook and a basal body. With the eukaryotes, they don't have a hook. So just filament and the basal body that's going to be anchored in the cell membrane. Okay, and the structure changes a little bit down in the basal body. It has a 9 plus 0 structure, meaning it actually changes into 9 triplets and 0 microtubules in the center. So I'm not sure the significance of that structural change, but it's noted there. Okay. The motion is also very different. So how do bacterial flagella move? They rotate, all right? They spin around and around and around. Eukaryotic flagella are always going to move in a whip-like motion. And sometimes it's really hard when you see something in full speed to be able to notice the difference. Let's see if I can open this. Whoops. I don't know why I have to figure out how. Sorry about that. I don't know. My iPad keeps doing that, and I'm not sure. Oh, full screen is unavailable. That's why. Okay. Well, this is like a 10-minute video, and sometimes whatever you can look at it, but it's showing you Euglena moving around, and it's kind of fascinating because a lot of times the Euglena, the flagella will stick out, and it'll just be the very end that's kind of whipping around, and it's moving towards, like moving in this direction, which I don't really understand the mechanics of how that works, but it's kind of fun to watch them move around. This video contrasts bacteria with sperm, and it shows the sperm flagella moving. So I would encourage you, don't memorize that bacteria move in rotation and eukaryotes move in a whip-like way. You should just be able to reason it out, because cilia, so another structure that we'll talk about next, have the same structure as flagella. They're made of that protein flagellin. And if a paramecium like this, if those cilia all moved around and around, like it would just be tumbling around. It wouldn't move in a purposeful way. Okay, so all of those are kind of beating in a whip, so it kind of moves it in a kind of purposeful way, I guess. So a little more about cilia. This is another method of locomotion for some of the protozoa. Some of our cells are ciliated as well. Okay, so again, let me write this down here. Some protozoa, some types of animal cells. Okay, we have ciliated cells in our respiratory tract. Okay, so again, if those cilia were moving around in circles, that would make sense. Okay, the purpose of them is to move like dust and things out of our lungs. Okay, move it in one direction. Again, there's only cilia. You will only find cilia on eukaryotic cells. Bacteria have some shorter kind of appendages. Remember, we called fimbriae or pili, but those were not functioning in locomotion. Those were functioning in mostly attachments. Okay, so, all right, they're shorter, they're stiffer than flagella, and there's going to be lots of them all around the surface. Cytoskeleton. I'm just going to mention this briefly. You can read more about it in your book if you're interested. The cytoskeleton is something that bacteria have as well, but it's usually much more pronounced in eukaryotic cells. And the cytoskeleton is considered to be composed of microtubules, microfilaments. That's what that middle picture is, and intermediate filaments. Okay, those are 3 different structures that are all considered part of the cytoskeleton. Basically, the cytoskeleton, what does it do? Provide structural support. And it's also kind of a network for transport. All right, so basically, for example, when a eukaryotic cell does maybe phagocytose some bacteria or whatever, it's going to enclose it in a membrane. That membrane doesn't just float anywhere in the cell, it's moved to specific places with the help of some type of cytoskeleton. Okay, and it's going to be, exit the cell again via some type of cytoskeleton. Cell biologists really like to use fluorescence microscopy here in order to see the cytoskeleton. We have fluorescent dyes that we can tag to either the tubulin or the actin filaments that show up and give us this really, really cool pictures. Okay. So then all those organelles that you find inside a eukaryotic cell, just kind of the basics of what it does. So again, if you're interested in this, there's lots more, you can read more about this in OpenStax, and I think I asked a few homework questions about it. But basically, of course, the nucleus, that's where the DNA is. Got a couple membranes around it. The endoplasmic reticulum, that's either considered the rough ER or the smooth ER. The rough ER is going to be studded with ribosomes. What kind of proteins do those ribosomes make? Ones that have to be transported maybe to the cell surface or out of the cell. So like one of our cells, like a B cell that makes antibodies, for instance, all those antibodies are made on the rough ER, and then they're going to be secreted outside of the cell. The smooth ER, excuse me, has lots of different functions. Of course, it doesn't have ribosomes. One of its more famous functions is lipid synthesis. So we'll make a note of that. The Golgi complex, just transport modification. So it encloses maybe proteins that are made in the rough ER and kind of escorts them wherever they need to go in the cell. It might, what kind of modifications might be made? Well, it might say add a sugar to a protein. So it makes it a glycoprotein, or might add a sugar to a lipid to make it a glycolipid. That's the kind of modifications that take place. And then the mitochondria and the chloroplasts, of course, the mitochondria is called the powerhouse of the cell. That's where a lot of steps of metabolism, most of our ATP synthesis happens there. And the chloroplast is where light is harvested for photosynthesis in a photosynthetic eukaryotic cell. Okay, we're going to come back to those in a minute. I also want to talk about the lysosomes in a little bit more detail, because again, we'll be talking about this a lot more when we talk about phagocytosis. But say one of our white blood cells is phagocytizing a bacteria. How is that bacteria digested? Well, it's the lysosomes that fuse with that endosome. Within the lysosome are lots and lots of digestive enzymes that most of the time is going to bring about the death of whatever was, if it was a bacteria, for instance, that was endocytosed. Okay, so lysosomes, again, lots of digestive enzymes in there. And there's some other organelles again that they talk about in your book that you don't need to worry about. Okay, I want to lastly come back to the mitochondria and chloroplasts a little bit, because it's through close study of those that we have this theory of how kind of the first eukaryotic cell came to be. So it's called the endosymbiotic theory. And again, a couple things that explains, or it's a theory, we can't prove it because it's a creation theory, right? Explains how eukaryotic cells, we'll say, came to be. It also, the theory also says that mitochondria and chloroplasts originated as bacteria. Okay, so what do I mean by those two things? Well, this is a theory that Lynn Margolis proposed in the 1960s. And when she first wrote this up into a paper, she was like laughed out of town. Like nobody would publish her. She was ridiculed by her colleagues. They said this was ridiculous. But over the next 20 years or so, all this data started to accumulate that really pointed towards she really was onto something. So there's lots of support for that today. Basically, she said back in the early soup of, you know, when life was originating, there was some anaerobic prokaryotic cell that engulfed an aerobic prokaryotic cell. And instead of eating that cell, like you would think that it would for nutrients, that second aerobic cell basically was able to live inside the other cell. So we call that endosymbiosis, when one organism lives inside another organism. So that's kind of, you know, the first eukaryotic cell, and probably that happened maybe several different times, you know, and this aerobic prokaryote became a mitochondria in one case, and maybe it became a chloroplast in another case. I don't know how the nucleus suddenly appeared, but that's kind of our best hypothesis for how cells got to be more complex. All right, so what's some support for this theory? Well, the mitochondria and chloroplasts, they are both about the size of a bacteria. Okay, and remember we said about 1 micron. Oops, forgot to put my 1. About 1 micron in diameter. So maybe that's coincidence, okay? But mitochondria and chloroplasts about the same size as a bacteria. Both these organelles replicate independently of the cell. And they actually reproduce themselves more like a bacteria. They more reproduce like binary fission, where 1 mitochondria kind of becomes 2, rather than mitosis, the way the eukaryotic cell is going to replicate its DNA. They each have their own DNA. Okay, and it's circular, so it's of the form that's more like what we see in prokaryotes. Now, most of that DNA, we think, eventually got moved to the nucleus. Okay, so that maybe doesn't support the theory, but they have, again, DNA that maybe at one time was able to replicate itself. And in my mind, probably the biggest support here is that both mitochondria and chloroplasts have 70 S ribosomes. Okay, so they have the ribosomes that we normally only see in prokaryotic cells. So again, it's not something that we can prove for sure, but it's a fairly accepted theory now that that's probably right on. Okay, so that ends the material for exam 1. Question?
So when cells replicate, do they replicate in them already? Like how does that transfer?
Yeah, so when the cells replicate, how exactly does that work? That's a good question. I think now they are kind of replicated more, you know, on the schedule with the cell rather than doing their own thing. But it seems like yes, because we think a lot of the DNA has been somehow moved to the nucleus. Yeah. Okay, good. No question.
Do the ribosomes and mitochondria and chloroplasts are they made of the same components as prokaryotic ribosomes?
Yes. So they are prokaryotic ribosomes. So when we take an antibiotic that targets prokaryotic ribosomes, some of the side effects that we can get is because it's targeting our mitochondria. So good. Okay, good. Well, we'll go ahead and move on. Again, even though this material now is pushed off to the next exam. Okay. So survey of eukaryotes. So we're going to spend a little time thinking about, you know, again, most of this class we're focused on bacteria, but we want to spend a little time thinking about microscopic eukaryotic organisms. And I've referred to some of them already just this lecture, but that can include fungi like the yeast here, different kinds of protozoa, down here, different kinds of algae, so freshwater algae. These here are diatoms, those are marine algae. Okay, so these are all, again, we're most interested in the single-celled eukaryotic cells. So we're going to talk a little bit about the protists and protozoa here to begin with. We'll tease apart those terms, protist, protozoa, what's the difference, algae. A few structures, so only a little bit of terminology, and we're going to have an example of a protozoa that causes disease, malaria. Mostly I threw that in just because it's really very interesting. And otherwise, protozoa can be a little bit dull. Fungi, when we think about a little bit more different structures of fungi, why do we study that? And we'll also take a look at an example of a parasitic fungi, Coccidioides species. All right, so that's where we're going. Classification. So I pulled this figure from our old textbook. Your textbook has a little different figure, a little different way of looking at this. So basically when we first started classification, remember Linnaeus said, hey, there's 2 kingdoms, plants and animals. We've been making that a little bit more complex ever since. Late, 1900s, we had a few more kingdoms, including the fungi and protists in terms of just the eukaryotes. Today, this is the actual accepted taxonomy that scientists use. So there's many different kingdoms here. The problem is with this, using the ribosomal RNA sequencing, is that nobody recognizes any of those kingdom names except for people that actually study those kinds of organisms. So we're going to kind of stick with some of these late 20th century designations, like using the term protists and protozoa. But just keep in mind that there are some more organisms that don't fit exactly right in that classification. Like this kingdom here, Stromenopelia, those are organisms that caused the Great Potato Blight in Ireland back in the 1800s. Those are really weird organisms called water molds. And they are described as algal protists that act like fungi. Okay, so they just don't fit right exactly into anything. Okay, so within this group, the protists, okay, that's going to include the protozoa, which are animal-like. That's going to include the algae, that are, of course, more plant-like, and there is a difference between algae and plants. It's not just that algae are little plants. And that includes this group called the water molds that we're not going to talk about anymore, but they are a little bit more, they act a little bit more like fungi. Okay, so that's a group of the protists. Okay, so it's a little confusing. I think a lot of people would use the term protist and protozoa kind of interchangeably, but technically the protozoa, again, are protists that act more like animals. Okay, so we're going to spend a little bit of time on the protozoa. Okay, if we thought about the characteristics of the protozoa, of course, they're eukaryotes, they're single-celled. Most of them are motile, and we used to kind of put them in groups based on their motility, whether they used a flagella, whether they used cilia, or whether they moved in amoeboid motion. They are not photosynthetic. Okay, so remember we called these, kind of the animal-like protists. So they're not photosynthetic. Some other protists are, but not that group called protozoa. And they do not have a cell wall. Now, some of them have something called a pellicle, which is a kind of a proteinaceous layer just inside the cell membrane. You might think it would be on the outside, but it's actually on the inside. Okay, so some of them have this pellicle just to kind of strengthen up their outer layer, I guess, and provide a little bit of rigidity. Okay, so here's a couple just examples of protozoa. Trypanosomes, one of the parasitic ones that cause disease. Incidentally, when protozoa cause disease, we correctly call them parasites because they're a eukaryotic pathogen. If a bacteria or a virus causes a disease, then we call them pathogens. Okay. Again, sometimes those words are used a little interchangeably as well. Okay, so trypanosomes, here's another one, Giardia, has a weird kind of, those eyes are not eyes, those are two nuclei. It's hard to make that out on that picture there. Giardia is the reason that you don't want to drink water from that pristine mountain stream, no matter how clear it looks, because if you get some Giardia in there, it's going to give you a bad case of diarrhea. Okay, so lots of free living protozoa. I'm kind of more interested in the parasitic ones, so we'll talk a little bit more about that. A lot of protozoa have very complicated life cycles. So we'll see that when we look at malaria. But what structures, there's a couple things that we could say, okay, they all have this in common. All protozoa are going to exist as a trophozoite. And that just means a motile feeding stage. Okay, so in this example here, I pulled off the net somewhere. That is an organism called Naegleria fowleri. And you probably haven't heard of that, but that's the one that's very rare, but people can be swimming and it will enter their nose and cause this really nasty amoebic meningoencephalitis. Potentially fatal infection. So very rare, but that organism actually has two different trophozoite stages. It can exist as an amoeba and also in a flagellated form. I think it's flagellated in the water, and then when it enters our body, it's in the amoeboid form. But those are the motile feeding stages. And then some protozoa can also exist as a cyst. And that's a hardy resting stage. Okay, so not going to be as hardy as, say, an endospore, a bacterial endospore, but again can persist, during times, when maybe nutrients aren't available, or yeah, something like that. Okay, so again, here's some scanning electron micrographs of Giardia, just an example of that trophozoite stage. This is in a gerbil intestine here, lots of Giardia just swimming around and feeding. Okay, so let's talk a little bit about malaria as an example of a parasitic protozoa. I like to talk about this because about half of the countries in the world, you can potentially get malaria when you're visiting those countries. So it's the single biggest threat to Americans when they travel in terms of, getting sick or getting seriously sick. There's something like 438,000 deaths worldwide. Most every year, most of those are in Africa. So this is one of the biggest threats, again, in terms of infectious disease. We used to have it here in the US, and it was eliminated like late 1800s. We could have it again. Those mosquitoes that carry it are present here, especially in the southern US, and it could become a problem again. So that's kind of a scary thought. So what is it caused by? 5 different species of Plasmodium. Okay, now you don't have to memorize these, but Plasmodium vivax, Plasmodium malariae, falciparum, ovule, and molessi. Those are the 5 different species that cause malaria. And this is just a blow up of that Plasmodium protozoa. Almost looks like a little teeny tiny worm. It's microscopic. The most serious kind of malaria is caused by the Plasmodium falciparum. And that's the kind that you would contract probably if you were in sub-Saharan Africa. That's the most serious. And then the next most serious is the Plasmodium vivax. That is more common in like Latin and South America and parts of Asia. So there's a little difference in terms of, how long it takes for you to contract the disease and what the disease looks like, depending on which of the species that you are infected with. But all of these Plasmodium protozoa are carried by a certain kind of mosquito, ones in the genus Anopheles. Okay, so Anopheles mosquitoes, and it's the female ones that transmit the protozoa because it's only the female mosquitoes that are taking blood that are biting people. Okay, so they need that blood to nourish their eggs. And the female mosquitoes, unfortunately, are not harmed at all. If they were, then we probably wouldn't have a problem. But the Plasmodium have part of their life cycle in the mosquito, and then they transfer back and forth between the humans and the mosquito. It's a cyclic disease. So typically attacks last maybe 6 to 10 hours and then are repeated maybe every 2 or 3 days. So you go through a cycle. If you're ill, a cold stage, a hot stage, and a sweating stage. So in the cold stage, it might last, just a few hours. The patient is shivering. They've got chills. And then in the hot stage, they suddenly spike a fever, pretty high fever, like 102, 103, 104, 105. Okay, really high fevers, headache, vomiting. Rapid faint pulse. Okay, so I've heard it described as being excruciatingly painful when you're in that hot stage, and people just they ache so much and they're so miserable they just want to die. But then eventually the fever breaks, and in that sweating stage, again, like you would think, you have profuse sweating and extreme fatigue. Lots of complications of this. Sometimes people's kidneys shut down. Of course, anemia, because as we'll see, this is targeting a patient's red blood cells. There can be neurological complications. So it's really a tough one. I won't get into the treatment of it. It is treatable, but it's not easily treatable. There's a lot of resistance to it. And the drugs that we take to keep us from getting it sometimes make us sick. So, if you're going on a big trip and you're on this malaria medication and it's making you sick, sometimes people quit taking it. And then, of course, then they're more susceptible to it. Okay, so we try mostly to just, control the mosquitoes, and, people sleep under mosquito nets and that sort of thing, but that's not foolproof. So if we look at the disease, this is a very complicated cycle of disease here. And I just want to, this is the diagram that the CDC gives. And I just want to point out a few things that I want you to understand about what's actually happening when patients get sick. So if you start right here, where the mosquito takes a blood meal. So a mosquito that is infected with the Plasmodium bites a human. What happens first? There's a progression of maturation of that Plasmodium that has to take place in the liver. I think it's in the hepatic vein. So we call that the human liver stage. Sometimes it's referred to as the exoerythrocyte cycle, so not in the red blood cell. Okay, but there's some maturation in the liver. Don't worry about kind of like the schizotes and all the different forms that we have for the actual Plasmodium. But when that maturation takes place of the Plasmodium, then it enters into the human blood stages, okay? The erythrocytic cycle. This is in human red blood cells. And this is where all the disease manifestations now are taking place. That cyclic, the chills and the fever and all that. That's where damage is actually being done to the red blood cells. This is the trophozoite stage. This is where you have the motile feeding stage of the Plasmodium. And now if a patient is bit by a mosquito, now the mosquito can take some of that Plasmodium with it. And then within the mosquito, that's over here on the left portion here. Now we have, again, maturation that takes place there. Don't worry about all the different cycles there, but there is a cyst stage, I call it oocyst, where it kind of hangs out for a while. Okay, so complicated. It took, we've had malaria for thousands of years, and we didn't elucidate, what was actually causing it until like the early 1900s. So complicated disease. We do have a vaccine for it now that's fairly new, but it's being given to children and saving lots of lives. So we'll talk about that more when we talk about vaccinations. Just kind of for fun here, if you think about the world's deadliest animal, this is on the Gates websites, I think. You think of things like lions and hippopotamuses, they're pretty mean, right? Well, if you think of the number of people on average every year that are killed by some of these animals, this is sort of surprising. Dogs, 25,000 people killed by dogs because they carry rabies, really. But snakes, lots of people killed by snake bites. And of course, humans kill lots of other people. But all in all, the most dangerous animal could be considered the mosquito. Mosquitoes transmit lots, and I think there's like 14 different infections, most of them viral. So if malaria is causing maybe 430,000 deaths, there's lots of other things like yellow fever and dengue and West Nile virus and Eastern equine encephalitis, lots of other things. So we could get rid of the mosquito and have a lot less disease, but that probably won't happen anytime soon. Okay, just let me finish up here talking about the algal protists. Just one slide here. Of course, algae are super important to the world's food chains, and we get a lot of oxygen from them, but they really don't cause disease unless you have maybe a red tide or something like that when we have an overgrowth of them. But so for the most part, don't cause disease, so we're not going to worry about them too much. Characteristics, they're eukaryotic, they're photosynthetic. They can be unicellular, multicellular, or clonal. Okay, so you can have unicellular algae like those diatoms in the middle picture there. There's giant kelp that stretches for miles in the oceans. That's technically algae. This is a picture of red algae up here. That's where we get agar. Remember, agar is what we add to media to solidify it. The Volvox, that's an example of a colonial algae, like single cells that kind of live together. So what's the difference? Oh, we won't get into structures here because there's so much variation. What's the difference between algae and plants? If you're interested, for algae, every cell in the reproductive structure can become a gamete, and that's not so with plants. So you can't really look at a plant or algae and know for sure. You have to kind of know something about its reproduction to know whether it's technically an algae or a plant. So just sort of FYI in case you ever wondered. And what about plankton? What kind of organism is plankton? Well, it turns out that plankton are just unicellular creatures that live in large bodies of water. So a plankton could be a bacteria, it could be an archaea, it could be a fungi, it could be a protozoa. We don't really know. So in case you wondered. We got further than I thought I would today. So I think we'll stop there. If you have any questions, hang around, make sure you get going on your homework. And I hope you have a good day.
Oh my God.
Yeah.
I was just reviewing my